Now let's take a look at how mitosis would occur in plant cells. In these images, we can see plant cells undergoing cell division by mitosis. We're reminded that these are plant cells because we can see the nice cell walls giving them their well-defined shape, and remembering that plant cells are packed in very tightly as they form plant tissues. Now, in this particular portion, we can see that the nuclear envelope has already begun to erode, and our chromosomes are forming as the chromatin condenses to form the short condensed chromosomes. We can see the nucleolus is disappearing, so this is a plant cell that is in prophase. As the chromosomes continue to form, they attach themselves to the mitotic spindle fibers and align at the equator of the cell during metaphase. During anaphase, the sister chromatids are pulled apart by the centromeres, allowing the chromatid arms to sort of trail behind as they move towards the opposite poles of the cell. And during telophase, we see the two identical daughter nuclei begin to reform. Now, this is where one of the major differences occurs, because in a plant cell, instead of having cytokinesis occur with cleavage, forming a cleavage furrow, we see the formation of the cell plate that grows across, splitting the cytoplasm of one cell from the other. And if we look at this in a diagrammatic view here at the bottom, we can see that vesicles from the Golgi complex will align themselves and congregate here, forming this cell plate. Not only does the cell plate, as it grows across splitting the cells during cytokinesis, produce new cell membrane, it will also produce the basic components like cellulose to form the cell wall. And here we can see this transmission electron microscope image showing the same thing. We can see these vesicles forming the cell plate here during telophase, showing us how cytokinesis occurs in these plant cells. Now, in this image, I want you to use this image to practice, right? You should be able to look at each of these plant cells and determine what phase of the cell cycle the cell is in. So use this to quiz yourself or quiz each other if you're working with a study partner. So I'll just go through some examples for you. So notice here in this cell, we have a nice well-defined circular nucleus, a dark nucleolus, and you can see this sort of grainy or granular genetic material making up the chromatin. This is a cell that is in interphase. Following interphase, the chromatin will condense to form the chromosomes, and the nuclear envelope will begin to erode away. And remember that the nucleus now that will begin to take on this sort of oval appearance. The nucleolus will leave, and this is a cell that is in prophase. Following prophase, the replicated chromosomes will align themselves at the equator of the cell during metaphase, their centromeres attached to the spindle fibers. And following metaphase, we see the sister chromatids getting pulled apart by their centromeres, moving to opposite poles of the cell during anaphase. Following anaphase, we see two daughter nuclei that are beginning to reform, and if we look very close here, we can even see the beginnings of the cell plate for cytokinesis during telophase.